thrilled to be here in Cambridge. I'm going to be here through the middle of August, and I'm hoping to make it down to London at least once or twice during that time. I can already tell you that the highlight of this trip is going to be the conversations that I have. So please, if you find anything that I say interesting, feel free to reach out. I'm also doing a lot of stuff on amyloid aggregation, which I know is a big area of research around here. And so with that, let me get started. And of course, the first thing on my slide is where I'm from. This is the plains of Kansas. And now let's move on. So before I start talking about phase separation, I want to take a slide here to talk about perspective. And I want to make a point here by comparing these two protein systems here. There's some noise going on with the window right now. Let me know if that becomes objectionable and I can close the window. So as a condensed matter physicist, I look at these two molecules, and the first thing that I start lining up here are the similarities. I'm going to look at them and I'm going to say, hey, these are both sort of small polypeptide chains. They're globular. They are weakly charged, if I'm going to look at some sort of colloid type of stability there. And they have, relatively similar folds to them. And so again, from my condensed matter physics point of view, I'm going to say with a fair amount of confidence here that anything I say about one of these molecules should apply equally well to the other. And any changes between them are just changing parameters like the charge or the radius. This conclusion is absolutely useless from the biological point of view because biology, you're interested in function. And the function of these two molecules is massively different. Ubiquitin is a molecule that signals a protein for degradation, whereas Barnase has an RNA cleavage function to it. And so this is going to be a theme that's going to pop up over and over again in this talk. And so hold this idea in your head. And with that, we'll move into these membraneless organelles. And Thomas did a wonderful job of introducing these. So I won't belabor this other than the fact that they occur throughout the cell. And presumably the function of every one of these things is different. Okay, but again, from the physics point of view, there's a lot of things in here that are similar. And they seem to be very similar to liquid phase separation. And so let's take and they have liquid properties. And so let's take a little diversion into liquid phase separation. And the first thing to point out here is that liquid phase separation is not a new phenomenon. We've known about this in protein systems as long as I've been alive. But the point I want to make about this is look at the conditions at which this is happening. Okay, this is lysozyme, which is famous for being an exceptionally easy protein to send through this. And, when you're at, sort of physiological salt concentrations down here, you're at massively unphysiological temperatures. When you start getting up to physiological temperatures, you're at massively unrealistic salt concentrations. And again, lysozyme is really easy to do with this. This is another example here. This is an antibody here. And the phase of separation coexistence window here is several degrees below freezing. Okay, and they had to do several extrapolation tricks in order to even identify this. And so based on the strength of nonspecific protein-protein interactions, this is sort of typically what you expect here. Phase separation is not really a physiological phenomenon. Okay, well, now that we're seeing these in things in cells, the question is how is this different than what we've known about lysozyme and antibodies and crystallins for all these years? And one of the big differences that emerges is that it's driven by polymer-like models. And in some cases, these polymers look like beaded necklaces of specifically folded domains that specifically recognize targets. In other cases, they're intrinsically disordered regions with side chains that attract each other. And so with this insight here, we can use well-established polymer theories here. For instance, Flory-Huggins, which again, Thomas did a wonderful job of introducing here. And what have we learned by that? Well, what we learned is that these polymer characteristics give a mechanism which we can tune this coexistence window into physiological regimes, okay? So we don't have a lot of control in these protein systems over how strong the interactions are. although there's caveats to that, but hopefully we'll address those later in the talk here. But just by changing the length of the polymer here, we can shift this coexistence window and we can move it into the physiological range. We can tune that right to the limit where the cell can conceivably turn it on and off. They can have some regulation into it. And so this is provides a lot of explanatory power as how this is different from what's been studied decades before. Another observation is that these long polymer-like structures aren't universally sticky, like the homopolymers that Florey Huggins was designed to interrogate. What you tend to happen are these discrete sort of sticky regimes separated by these long inert spacers. And again, we have good theories for describing this, pioneered by Semenov and Rubinstein. And again, this provides a mechanism for which these cells can use in order to tune the coexistence window. Are my windows blocking everything, or can everybody see my entire slide? Okay. But I'd argue that the other insight of this is that you don't, by having this sort of architecture, you also have these big sections of the chain here that are not involved in phase separation, and those can play other functions as well. So what I really want to tell you about here is two stories here that we've looked at in my group here. And in both of these cases these are binary mixtures of polymer-like models that phase separate when you mix the two ingredients together. And both of these have all these sort of hallmark characteristics of these sticker and spacer polymer systems here. So let me introduce these two systems here. In each case, you have one component that is composed of folded domains, okay? And so these are folded domains that have discrete binding sites. In the SPOP case here, there are these little green domains that hang off the side, and these SUMO domains are linked together, and these are, binding domains. And in both cases, these binding domains specifically recognize an intrinsically disordered partner. So over here on the left, our binding partner for SPOP is a molecule called DAX, and I've labeled sort of these lengths in here and highlighted the binding domains. And polysim SIM stands for sumo interacting motif. So these yellow things are just the sequence that these blue things like to bind to. Okay, so now let's talk about some differences here. Okay, so the first thing we can look at is the valence. How many of these sticky domains do we have? SPOP polymerizes into rods that are essentially rigid on the length scales that we're interested in. Under the conditions I'm going to talk about, the polymerization length is on the order of 50. In DAX, we think that these two pairs essentially act cooperatively. And so the effective valence of this thing is 2. You have these 2 pairs of dimers here. Over here on the left, SUMO and SIM is a synthetic system, which I'll talk upon a little bit more in a little bit more detail later. And so both of these have been engineered to have a valence of 10. If we talk about the spacers here, again, the spacer on SPOP is essentially 0 because these are rigid rods. The spacer between our two interacting motifs here is 67 amino acids. And again, we have an engineered system, so it's symmetric. There's a flexible amino acid link, or about 12 amino acids, or exactly 12 amino acids between each sticker. Okay, now, the interesting thing about these systems is that in each case, we looked at it and said, there's something a little weird here. And in the case of the SPOP DAX system, you have a very interesting phase diagram here. So think what we're looking at here is we're looking at the association of SPOP here, and we're going to think of DAX as a cross-linking molecule. Okay, so DAX is going to be something that pulls these essentially repulsive SPOPs together. And so think of this as sort of your interaction strength axis here. Okay, so if I have 0 interaction strength, I basically have a dilute vapor of SPOP rods. If I add a little bit of sticky DAX here, I create these sort of filamentous bundles up here. And if I keep adding DAX, then I get these liquid droplets down here. One more detail I'm going to throw in that I'll discuss later here is that if you go to very high DAX concentrations, you can get a DAX liquid that doesn't have any SPOP involved here. But to me, the reason this caught my eye is because this is effectively an inversion of the phase diagram here. Usually you would think that as you increase your interaction strength, you would go from vapor to liquid to solid. And here you're essentially going from vapor to solid to liquid. So this flip of the liquid and the solid phases was real interesting to me. So after playing with a lot of different models for this, which I'm not going to go into, here's the model that ended up working here. Okay, so what we have here in the gel phase here is not surprising. You've got a bunch of these rigid rods that are held together by these bivalent cross-linkers here. Over here, down here in the vapor phase, it's essentially the same story, except you just don't have enough of these cross links to overcome the entropic penalty to bring them together. So you remain in the vapor phase. But the interesting story is when you move out here into the concentrated regime here, and what happens is you saturate all the binding sites on these rods here. And what that means is that you can't form any cross links because the other head of this guy here doesn't have any available binding sites there. But remember I said that DAX phase separates by itself. So what you're essentially doing is you're adding up a bunch of these weak DAX-DAX interactions here in order to bring this phase separation regime for DAX down to much lower concentrations here. Now, one of the things I did over the pandemic was convert all my statistical mechanics models into Lego videos. And so because I have this, here's the Lego version of what I just said. Here's your rigid gel state here. And here's your liquid state of these sort of, think of these as cylindrical brushes. And okay, so our collaborators on this project, this is Tanya Matog's group at St. Jude. They went and measured the parameters in the model, which are essentially the affinity constants for bivalent and monovalent interaction of DAX here. And with those experiments, the parameter space is extremely restricted here. We only have a couple little small things to play with there and within very tight ranges. And with that, we can reproduce the phase diagram very well. And in fact, we can get the concentration profiles within the condensed phase. And there's no free parameters in this model here at all. And there's interesting stories there that I'm going to skip in the interest of time. Okay. So to wrap up this story, and again, there's a lot of interesting physics here I'm skipping. The take-home message is here is that you have a gel phase that's stabilized by these strong bivalent cross-linking interactions. You've got a liquid phase that is driven by these sort of weaker DAX-DAX interactions. These are the interactions between your brushes. And the transition between these two phases is something that you can drive at equilibrium just by changing the relative stoichiometry of the rods to the bristles. Okay, so with that, let me move into my other story here. This is the Sumo Sim story. And as I mentioned, this is an engineered system, and it was engineered in order to study the physics of scaffold client behavior. And this is something that, again, Thomas gratefully introduced for me. And so the idea, this is Mike Rosen at UT Southwestern, was that we were going to use these polysumo and polysim molecules as a scaffold in order to build a phase separated compartment. And then we're going to see, investigate the physics of how this compartment can recruit client molecules. And so for their clients, what they did is they just use sort of monovalent versions of the same domains here, and then they fluorescently tagged them so that they could read out the recruitment of them. Okay, so we started off with them. And again, we thought, okay, this is a straightforward application of Flory-Huggins. We've got our SPOP DAX model. We got the associative polymers model of Semenov and Rubinstein. And so all we need to do is pull a model off the shelf, plug it in, and see how it works. And to cut a long story short, this failed spectacularly. And in our theories, no matter which model we used, predicted basically closed packed conformations. And of course, these are theorist units here. So this is 90% of some lattice model here of a characteristic molecular size. But no matter how you scale this back to real units, you're at least an order of magnitude larger than the concentrations that were observed in the experiments. And after trying every version of trying to fix this, we were forced to reject this model. So this is clearly not what's happening. Okay, and this makes sense because what you have here is you have a characteristic length scale that's coming out here from the spacing between adjacent domains, okay? And so if you're going to form a random network, you've got some sort of mesh work here. And so the size of your mesh is your characteristic length scale here, and that's being set by this spacing between your binding domains. Okay, but if you think about it, this is a poor way to satisfy all these binding domains. In fact, a far more efficient way to do it would be, say, if I line these molecules up adjacent to each other. So instead of doing this, if I do this. And so then I can sort of form a chain for here. This is much more efficient at satisfying these binding domains. Now, of course, this is a thermal system, and so that means we're going to generate defects. And we've got two types of defects. One are the sticky ends, and these are the things we use in order to propagate this filament. And then we have these gaps in between, where one blue molecule ends, one next blue molecule starts, or vice versa for the yellow ones. Okay, and so this defect spacing here introduces a new length scale into the system here. And so the argument here is that what we're doing here is we're actually creating a network here in which the mesh size is set by your defect spacing, which is on the order of the molecule length, not the domain spacing here. And this explains why your network is so much more diffuse than you would expect with a random polymer theory. And so what we need to do is calculate the defect density. But before we do that, I once again have to show off my pandemic play. And here's the Lego version of the same thing. So we're building these one-dimensional filaments with these two types of defects. And then what's going to happen is your filaments are going to assemble into a 3D network that's connected at the defects. Okay, so what we're going to do is we're going to use these client binding experiments that Mike Rosen did in order to test our theory. And conveniently, that's probing at exactly the same thing that our theory is getting at, which is calculating the partition function of these filaments, and we can tell how many defects there are. And there's lots of fun statistical mechanics that went into doing this that I'd be happy to answer in questions. But I'm going to skip in the interest of time. So here's the result. This is their experiment here. And what we're looking at here is the fluorescence of the clients as a function of your scaffold ratio. Okay, so here I've got more blue scaffolds, here I've got more yellow scaffolds. And as you would expect, if I have more blue scaffolds, I recruit more of the yellow clients. And so you see that down in this corner of this basically phase diagram. Now, the interesting thing is if you look along this bottom row quantitatively here, and let me do that quantitatively for you because the eye is not good at picking this up. And what you see is that there's this non-monotonic dependence on the client recruitment here. It gets stronger here and then it gets weaker out here. Okay, and that's what we want to understand. And to our great surprise, this ended up coming out of this, this one-dimensional filamentous model here. And here's how it works. So if I have a filament like this, and I am going to start increasing the concentrations of this blue species here, well, where can this blue species go? Well, it can go on this sticky end over here. And so when I attach another blue molecule to the end of this filament here, not surprisingly, I create these new spots where my yellow client can come along and bind to it. And so this increases my fluorescence in this direction here. Now, the problem is that when I continue to add my blue chains into the solution here, there's no more places where I can add the blue molecule to this filament. I've occupied all my sticky ends here. And so what happens is that my blue scaffolds start to accumulate in free solution. Okay, now these things aren't recruited to the condensate because they're off by themselves. And these guys here can compete for my clients, which pulls them back out of the condensate. And this results in the non-monotonicity. My client recruitment goes back down because it's being outcompeted in the dilute phase. Now compare this to what would happen if you were in a more random network. In this case, your ability to tolerate a scaffold asymmetry is much higher. And so you're still eventually going to get the non-monotonicity, but it's going to happen much later. You're going to have to go out to much higher stoichiometry asymmetries. Now, importantly, what this filamentous structure does is it creates correlations in where your available binding sites are. You're more likely to have two gaps next to each other than just sort of randomly distributed somewhere else in space. And what this does is it makes the system sensitive to the valence of the clients you have. Okay, so if you have a monovalent client, well, this sees the system differently than, say, a divalent client. And this creates different affinities for the different valences. And so this here is the theory compared to the experiments. The experiments have the error bars. This is for trivalent clients, bivalent clients, and monovalent clients. And notice, of course, the height of the peak changes, but importantly, the location of the peak changes. This one is peaked around 60. This one's peaked closer to 65. And this one's peaked closer to 75 or 80. Now, interestingly, because it's a theory, we can go in and we can tune the affinity of the clients here. And if we change the affinity of these clients, we can change the height of the peak, but it actually has very little effect on the location of the peak. So what this does is it gives us two different knobs here. We can adjust the location of the peak and we can adjust the height of the peak. And that leads us to this very interesting prediction here that you can tune your network here to specifically identify and recruit either this client or this client. So in this case, these are clients that have tuned affinities. This one's trivalent, this one's monovalent, and based on the state of the system, your network, which is liquid, I will remind you, can specifically recognize and recruit one client while rejecting another one. Okay, so let me summarize the Sumo Sim story. We have these filamentous assemblies. This causes a much lower density than we would otherwise expect. We have these gap and end defects, which are going to form crosslinks and also bring the clients in. And this network architecture allows a specific recognition and recruitment of clients. Now, something really interesting happens when you compare and contrast these two results here. Okay, remember, we went into this system expecting that we were just going to apply Flory-Huggins or stickers and spacers or something like that and plug things in. And instead, what we found is we found very different network architectures, and we couldn't get the modeling right until we put in this specific network architecture of the two different systems. Now, this network architecture, I would argue, is what you would want to design if you wanted to have control over the dynamic properties of your system. You can switch at equilibrium back and forth between a rigid, a non-dynamical system and a fluid dynamical system. Whereas this architecture, this is the one you want to have if you want to be able to specifically identify or reject one client versus the other. Okay, so what is the conclusion in this? Okay, well, the tongue-in-cheek way I describe this is that we went looking for physics universality here, and instead what we found is structure function. Okay, so coming back to the second slide here, we sort of like, is this the physics point of view or the biologist point of view? We went in looking for the physics point of view, and we instead found these specific structure function properties here. You know, I bet anything that evolution is going to exploit this. But let's step back a little bit and let's ask, are there any universal principles that come out of this? What is it that enables the system in order to have these specific properties here? Okay, so to do that, let's talk about a structure function. Of course, this is, you know, ubiquitous in biology. It arguably underlies everything. And so if I have a small, you know, enzyme here, its function comes from the structure it adopts. And we want that structure to be stable with respect to thermal fluctuations. And so that means that we need it to be stable on the order of 10 kT. Okay, that's the strength you need in order to overcome thermal fluctuations in order to allow this thing to be catalytically active. Okay, so let's take this thermal stability here and let's apply it to a three-dimensional network, something that's going to form a condensed phase. And the result is a rock. Okay, you've got something that is just rigidly held into place and it's stable. It's got a structure, but rocks are not alive. Okay, this is not, you're not going to get any biological function out of this. Okay, it's just sitting there. Okay, so we overdid it. Okay, let's dial down the strength of our interactions and let's get this thing to flow and adopt dynamical properties so things can come in and things can come out. Well, what's going to happen is you're going to weaken that down until it turns into a mush, but then you lose all your structure. And with all your structure, you've lost any specific function to it. And so the question is how do we overcome this sort of, you know, push-pull between these two competing factors? Well, we've got two examples of how that was done. The SPOP-DAX and the Sumo Sim, and we can look for what are the unifying principles there. And the answer is what they do is they mix these strong and the weak interactions together. What you do in each system is you build these structures out of strong interactions that are going to define the functionality of your system. And then what you want to do is you want to bring those strongly defined structures together with weaker interactions that can break and reform and flow with dynamical properties. And so this is what we're thinking is sort of a generic architecture for these condensed systems here, is that you're going to have multiple strengths of the interactions. The strong interactions are going to bring together these functional assemblies here. These are the things that are going to optimize one specific condensate to perform its specific function, whatever that might be in the cell. And then you're going to have weaker interactions to bring these assemblies together and gain whatever advantages you have out of compartmentalization or localization. And looking throughout the cell, we see this motif pop up everywhere, all over the place, any place where there's been any sort of detailed structural characterization of these condensed phases. Here we see it in the nucleolus, here we see it in chromatin, there's evidence of this in stress granules, you name it. We think that this is a universal feature. And so with that, I'll put up my acknowledgement slide. The people that worked on this project, Nelson Romello, Kamal, did all the Sumo Sim work, my collaborators, and I'd be happy to answer any questions.
Thanks a lot, Jeremy. It was fantastic. So let me first ask the audience, do you have any questions?
I have a question.
Hi, Rosanna.
Hi, Angela. Hi, Jeremy. Fantastic.
Hi.
Okay, that was super interesting. Yes, I was very interested about the last few slides that you presented, this idea of like the strong versus weak interactions, particularly in the context of chromatin.
Yeah.
Because there's been this idea that, for instance, these transcription factors actually are perhaps built in that way, where you have like DNA recognition motifs that bind preferentially to DNA sequences via globular domains, and then the IDRs of the proteins recruit like a different range of molecules to then start building these factories. But about nucleosomes, I was not really sure kind of what you meant. I'm sure it's super interesting. So I would like to hear what is the ideas of these nucleosomes and these soft versus weak interactions.
Yeah, so I think that I think chromatin is actually a fantastic example of this, and actually I think what what's going on with chromatin there is that it does have to switch states. You need to go from a compact state in order to a dilute state, depending on whether genes are on or off, whether you want a gene accessible or whether you want it packed away. So in some sense, it's got some of the characteristics there of the SPOP-DAC system. The SPOP-DAC system sort of goes between this sort of more densely packed state and the more dilute state. But I'm sure that the mechanism that DNA does this is completely different. And the way DNA does it, you've already touched upon this, is that you've got these nucleosomes that are essentially your stronger interactions, and the weaker interactions that cause it to condense are probably between the disordered tails of the nucleosomes. But that's only scratching the surface. And there's a lot more. How does that happen? How is this regulated? These are the types of questions that I think that there's. We'll talk about this a lot tomorrow morning, I'm sure. But,
okay.
But yeah, I think that's sort of a perfect example of what's going on here, and you don't get that sort of. You have to have the strong interactions. You have to have the weak interactions to condense things in order to make it reversible, and then you have to have control over it. And all of these are going to require these sorts of hierarchical motifs.
Okay, thanks. And a follow-up question, on the same kind of ideas. There seems to be a lot of proteins, the multi-domain ones that undergo phase separation that also oligomerize via these folded domains.
Yeah.
So do you think there's like a functional reason why they do that, or like an advantage for them to be able to? Actually, a lot of them are like found in dimers. I don't know why, but they do form these dimers via the globular domain.
No, I think it's exactly for this reason. In fact, just about anything that's been studied, in vitro, what they do is they try and identify the phase separated, phase separation driving regime, and then they cut that off and they study it in vitro, and over and over and over again, you find that it's undersaturated in the cells. Based on compare what's in vitro to in vivo, it's always apparently undersaturated in vivo. And the reason is that those non-driving regimes form these higher order interactions, things like dimerization and this. And so the prediction of our model here is that you basically never want one driving force to be responsible for everything. You can, that'll always work, okay? But there's always a way to make what you want to do better if you mix multiple interactions with it. And I've tried every negative control with me. I'm like, can I think of just a pure compartmentalization mechanism where hierarchical, I can always think of a counterexample. Nope, actually I could make the properties better if I mix some strong interactions with some weak interactions. And so I think evolution is always gonna, this is like, sperm egg divergence there, the middle ground just is evolutionarily useless. You can always optimize it by mixing the strong with the weak. And that's what the dimerization domains are doing. That's your strong interaction.
Can I ask a question? So
Yeah.
in this first, the Pop DAX story where you spoke like kind of like when they're the crosslinkers, when they're low densities, it's just a gas. And then when you increase it, they cross-link different molecules. But then when you have a lot of them, they kind of have dangling ends that stick together and make a liquid and not a solid. But like, do you ever, so I mean, you didn't speak much about the details, how you did these phase diagrams, but do you ever get them, instead of making a liquid, making kind of finite size aggregates, because they have these kind of saturated ends that, if they're not as sticky, they might want to have their entropy and make finite size things that might have a function, right? Because things might not want to only phase separate, but you might want to have a certain size.
Yeah. Okay. So good question. So what I showed you is just a mean field theory that we wrote down. So we're not going to get any finite size assembly out of that. What I currently have Tien in the group. He's down here. He's working on the surface tension of these assemblies. So he's sort of using lattice models of basically that exact free energy there. So no, we don't see any indication of what you're asking for there, asking about there. But that's a good point. We haven't thought about it either. So if we had seen it, I'm not sure we would have noticed. So that's an excellent question.